Sensory organs for hearing and balance in mammals and most other terrestrial vertebrates are both found in the ear. Stereocilia play a big role in our sense of hearing and balance by transmitting the signals to the brain. I'll start by going over how vibrations travel through the ear. You can follow along with the diagrams on the first page. Our perception of sound starts when someone talks or taps their foot. This creates percussion waves in the air. These waves travel into the ear through the auditory canal and hit the eardrum. The vibrations then pass through the three bones called the malleus, ingus, and stapes in the middle ear and hit a membrane. This creates pressure waves in the fluid of the cochlea before dissipating at a second membrane. It is much easier to see how this works by seeing the cochlea uncoil. But keep in mind that this cochlea can't actually uncoil. So after the vibrations from the stapes hit the first membrane, which is called the oval window, the pressure waves travel through the fluid inside the cochlea, first through the vestibular canal, then through the tympanic canal. When they hit the round window at the end, they dissipate, or disappear. The structure that the waves are moving around is called the organ of Corti. This is surrounded by the basilar membrane. So as the waves move through the vestibular and tympanic canals, marked BC and TC, it puts pressure on the basilar membrane in a certain area based on volume and pitch. Inside the basilar membrane is the organ of Corti. The pressure from the basilar membrane is put onto the tectorial membrane. Then the pressure from the tectorial membrane is put onto the stereocilia. This bends them one way or the other. Stereocilia are held in place by a hair cell, which are connected to axons of sensory neurons, which lead to the auditory nerve, which eventually leads to the brain. Now, before I go over how stereocilia actually transmit these signals, I'll quickly explain our sense of balance. Again, you can follow along with the diagrams that are on page 2. In the inner ear, we have the semicircular canals. There are 3 of them, and they are arranged in 3 different spatial planes. These detect angular movement or rotation of the head, like when you look left to right. There are also the utricle, and saccule. These detect our head's position in relation to gravity, like when you nod. The semicircular canals, utricle, and saccule all detect these movements in the same way. At the base of the canals, there are swellings, called ampullas. These contain a fluid called endolymph. The utricle and saccule also contain the same fluid. Since endolymph is a liquid, it cannot move with the head as it tilts. So as our head moves, it pushes the endolymph on a gelatinous cap called the cupula. So as their head moves one way, the endolymph flows the other way. This bends the cupula. Underneath the cupula are a bunch of hair cells. And these hair cells have stereocilia that get projected into the cupula. So therefore, when the cupula gets bent by the flow of endolymph, the stereocilia also get bent. Stereocilia can then take this movement and transmit it through the hair cells to the nerve fibers that eventually lead to the brain. Now that I've gone over how we hear and how we have our sense of balance, I'll go over what stereocilia are, what they're made of, and how they create these signals that our brain can interpret.
There is room on the 3rd sheet of paper for notes. Stereocilia are microsensing organelles of hair cells. They are typically 10 to 50 micrometers in length and are always arranged in bundles of 30 to 300. In these bundles, there are multiple rows that each increase in height, like a staircase. When the stereocilia get bent towards the tallest of them, this depolarizes the hair cell they are attached to, which ends up increasing the signal to the brain. When they are bent the other way, towards the shorter stereocilia, this hyperpolarizes the cell and will decrease the signal being sent. So through the hair cells, the stereocilia can turn mechanical energy from the bending into electrical signals that the brain can interpret. Stereocilia are made up of cytoplasm. They also have a rigid core that consists of cross-linked actin filaments, which can be up to 120 micrometers in length. Actin is one of the most abundant proteins in most eukaryotic cells. They have positive ends that are near the tips of the stereocilia and negative ends that are by the base. These actin filaments also anchor the stereocilia to the membrane of the hair cells. These are just some of the basics of what stereocilia are. There are a few differences between the stereocilia used in hearing and balance. In hearing, they are located in the organ of Corti. When they are used in balance, they are located in the utricle, saccule, and bases of the semicircular canals. There is one tallest stereocilium called the kinocilium, which is used in balance, but not in hearing. In hearing, the stereocilia have tip links. These are filamentous structures that connect the tips of the stereocilia. These tips are made of very fine filaments and act like springs. When they get stretched, it's the same as when they are depolarized. These tip links are also responsible for the maintenance of the bundles of stereocilia. In balance, in the utricle and saccule, the stereocilia are not oriented in a consistent direction. They are consistent, however, in the bases of the ampullas of the semicircular canals. So those are the differences between stereocilia used in two different ways. I hope this helped you learn about how stereocilia are used in hearing and balance. Thanks for watching.